hang on. Is this on? Yeah.
The
Is this, oh,
red light's on it.
red light's on. Yeah, you can hear me? Raise your hand if you can't hear me. Well, thank you very much for having me. It's a pleasure to be here. Thank you to Mike and Vincent. And thanks to Alan Rodrigo. Is Alan here for trading places with me? I was originally scheduled to speak on Thursday, but can't be here on Thursday. So Alan very kindly traded places with me. Be sure that you don't miss Alan's talk on Thursday. It will be very good. Okay, I'm going to tell you a little bit about our work on mixture modeling in phylogenetic inference. I should say up front that a lot of the work is very preliminary. There's a lot of sort of cherry picking here in that we're just seeing what mixture models can do. We've had a lot of luck with them. We think they're a very important way of going about characterizing gene sequence data. And I want to show you some of the things that we've come up with. Now, the talk, I'll first give you just a little bit of background before I tell you about formally what mixture models are. I'll give you just a little bit of background, and then the talk will really be divided into 3 sections, and if I can get through them all, I'll be exhausted, as you probably will be too. I'm going to tell you a little bit about mixture modeling on what we call pattern heterogeneity, in which we allow the gene sequence, different parts of the gene sequence, to evolve according to different models of evolution. I'll talk to you about a mixture model for heterotachy, that is, that a site can evolve at different rates in different parts of the tree. And then finally, I'll close with some mixtures on topologies, where we actually allow different topologies to fit the data. Okay, some background. This is really just for fun. Here's the latest slide from GenBank on the growth of GenBank. And you see it says 1982 to 2005. GenBank is growing so rapidly, I think even they are embarrassed at how straight up and down that graph goes. I downloaded that just a few days ago. And it's increasing, I think, at about 10 or 12 billion sequences or base pairs per annum now. This is really grist for Tandy's mill. This is why she can make her huge phylogenetic trees, or it's at least the stuff that goes into her programs. And there's a really fun thing about this. You can see there was a period of sort of hyperinflation right around the time of the release of the Human Genome Project, in which new gene sequences were appearing almost at an infinite rate. The other fun thing about this graph, though, just by way of background, is that look over here. This is the sequences, and this is base pairs, and they differ by a factor of 10 to the 3. Genes, open reading frames in nature, are about 1,000 base pairs long. This is a really strange sort of thing. It's true right across prokaryote and eukaryote genes. A typical gene is about 1,000 base pairs long. Nobody really knows why. Now, coming back down to Earth, we're not talking in terms of billions and billions and billions like Carl Sagan used to. But here's a growth in the use of molecular phylogenies in scientific research. This is off the Web of Science. If you just type in the key words molecular and phylogeny, and you have it search titles and abstracts and so on, this is the growth in the use of molecular phylogenies. I guess this is why we exist, because a lot of people are using molecular phylogenies. If you're an author writing a book on phylogenetic methods, it's not quite J.K. Rowling territory, but maybe you'll have 24,000 people buy your book. This curve used to be going up exponentially. I published a graph on this a few years ago. When it was down in here, it was actually going up exponentially, but you can see that the period doubling time, which used to be 2 years back around 1998, 1999, is now slowing down, but it's still going up pretty rapidly. And you can see there, by the end of this year, there should be around 25,000 published papers using molecular phylogenies. And I'm sure that's an underestimate. This is just a keyword search. Okay, that's just a bit of background, saying there's a lot of stuff for us to work on, a lot of gene sequences for us to analyze, and so on. All right, let's talk about mixture models in phylogenetic inference. And let me sort of define, if I can, what a mixture model is and how it differs from conventional inference. Now, without changing slides, let's see, is it that? Yeah. So if we take as our starting point in our usual sort of likelihood framework that we're interested in the likelihood of some model, which I'll call Q, meaning a kind of rate matrix, is proportional to the probability of the data given that model. And then we typically, in a conventional likelihood model, calculate the probability of the data given the model in some topology or tree. And that's just our usual assumptions, that it's the product over sites of the probability of the data given the model of evolution Q and the topology. Now, mixture modeling allows us, rather than to characterize the data with a single model, mixture modeling allows us to sum the likelihood at every site over more than one model of evolution. And so if we talk about a mixture model in the actual model of gene sequence evolution, we can talk about now calculating the probability of the data given a series of rate matrices, which we'll call Q. And what I'll have in mind here for the rest of the discussion of this model, this mixture model in Q, I call it, is a GTR rate matrix. So what we're going to do is find the likelihood of the data at each site summed over a series of rate matrices, summed over these J rate matrices. That is, at every site, we sum the likelihood over these models, weighted by some probability that the site conforms to that model of evolution. Okay, so it's the standard likelihood framework, but now we're allowing each site to be characterized by more than one model of evolution. That is, we don't partition the data. We simply sum each site over each of the models of evolution, and we estimate these weights empirically. Okay, we call this a pattern heterogeneity model because what it allows us to do is to estimate a number of different rate matrices on the data without partitioning the data or without telling the data beforehand how many rate matrices there are. All right, now just by way of contrast, and we'll get onto this later in the talk, we could define a mixture model if we say that the topology is some combination of a shape or a structure and a vector of branch lengths. We could define a mixture model on the branch lengths by calculating the probability of the data given some model of evolution, but now we're going to sum the likelihood at each site over a series of branch length sets, each one of which corresponds to the same topology, but we're allowing the branches to take different lengths for different sites. We'll call that a mixture model in T, and that'll be useful for investigating heterotachy, but we'll get onto that later. Okay, that gives you just a sense of what the pattern heterogeneity model is, and we're interested now in estimating these Qs and seeing if they can tell us something interesting about what's going on in the data. Okay, so an overview of phylogenetic inference from gene sequences. We usually start with an alignment. We then adopt some model of evolution, and we use that to infer a tree. Popular modifications to that basic homogeneous model are, that we can incorporate rate heterogeneity. That is, we might fit some sort of discrete gamma model across this rate matrix. Or investigators will frequently partition the data if they think different parts of the data follow different partitions. That is, they'll apply a different model of evolution to different parts of the data. Now, part of the message of this talk is that we don't need to do these things because mixture models do it for you. And it's also the case that we'll see that partitioning frequently leads to a worse result than just allowing a mixture model to decide on the best way to partition the data. And I use this word partition because, incorrectly, because in fact we're not going to partition the data with a mixture model. We're going to allow every site with some probability to be characterized by every model of evolution. Okay, so limitations to the usual homogeneous or rate heterogeneity or partitioning approaches, we've talked about them. Not all sites may evolve according to the same model. We adopt a gamma model, but rate variation may not be gamma. I think this is a very minor complaint, but that's just one thing we could think about. And partitioning the data presumes the investigator knows with certainty which model best applies to each site. And that we'll see is that that's just seldom the case. Okay, let's motivate this thing we call the pattern heterogeneity model of gene sequence evolution. Let's allow for different sites to evolve in quantitatively or qualitatively different ways. That is, the parameters of these GTR models of sequence evolution, we're going to allow them to diverge in any way they please. And so by qualitatively different, I don't just mean they're scalars of each other, as would be true of a rate heterogeneity model. And we're not going to partition in any way the data a priori. The method is we'll fit more than one model of evolution to each site and sum the likelihood as I described. The motivation is that this pattern heterogeneity model will always equal or better the performance of homogeneous models because the pattern heterogeneity model will simplify to a homogeneous model when that's all there is in the data, or I should say a single rate model. And it will always do as well as a gamma rate heterogeneity model because the pattern heterogeneity model could be a gamma rate heterogeneity model in the sense that our different models of evolution could adopt the gamma scaling. Okay, and we'll see that this model frequently yields substantial improvements in the log likelihood. Now, I should preface this by saying, as I did at the outset, that a lot of this work is preliminary. We haven't done the hard work of theorems or proofs, and we haven't done the hard work of ultra-massive simulations, but we're getting a sense of what these mixture models can give us. Okay, so some applications is that you might have a very long gene that has just more than one pattern of evolution in it. Different sites evolve differently. You might have a concatenated alignment with lots of genes, and the genes evolve differently, and so different models of evolution will characterize the different genes. Or you might have one of these supermatrix alignments, and different bits of this supermatrix are characterized by different models. All right, let's see if we can detect pattern heterogeneity in simulated data. What we did was we generated simulated sequence data on a random tree. Here it is, I think it was 50 tips. And we simulated sort of two genes, if you will, with different patterns of substitutions. And so we drew these at random, these two GTR rate matrices, and here are the rates in the black and in the blue. And we simulated data up that tree. I think it was 2,000 sites, 1,200 sites for the first rate matrix, and 800 sites for the second one. And then we analyzed those data with a homogeneous model and a pattern heterogeneity model, and I'll explain this more in a moment. And in all of our cases where we're estimating parameters, we're estimating the parameters from the posterior distributions that we derive from Markov chain Monte Carlo methods. Okay, so here's the time series plot of the posterior likelihoods for data simulated from these two qualitatively distinct rate matrices. Here are the two rate matrices here that generated these data. We fitted a gamma rate heterogeneity model and a single homogeneous GTR model, and you get a likelihood like that. They fall on top of each other because there isn't any rate heterogeneity in the data. And then we also fitted this pattern heterogeneity model, and we didn't tell it how many rate matrices were in the data. We would fit one rate matrix, two rate matrices, three, and we would test using Bayes factors how many rate matrices were required to explain the data. And we get this rather large increase in the likelihood, not surprisingly, for fitting two different rate matrices to the data. And we can go back here and look at the actual values of the input rate parameters that went into the two rate matrices. Here they are here. And get out here the mean of the posterior distributions of the rates that we got out of our Markov chain. And you see we do a pretty good job of retrieving those input rate matrices. And now this is by this process of summing the likelihood at each site over each of the rate matrices weighted by their estimated weight in the data. So we can retrieve the input rate parameters. We can go back to the data and we can ask at every site in the alignment, our 2,000 site long alignment, we can ask at every site, were you better characterized by Q1, the first rate matrix, or by Q2, the second rate matrix? Because remember, we're summing the likelihood at each site over both of them. We can ask posteriorly, which one fits you better? Are you more likely to have come from rate matrix 1 or rate matrix 2? And you see we get this very clear separation here. For the first 1,200, you're better characterized by the first rate matrix, and then it drops. And for the latter 800, you're characterized better by the second rate matrix. So it just shows that the information is in the data to identify these rate matrices. And we didn't tell the method to look there, to chop it at 1,200. Okay, so it looks as if we can identify these unknown matrices, and it looks as if we can identify the regions of the gene that are best served by the different matrices in this pattern heterogeneity model. Now, can we detect rate heterogeneity in simulated data? Well, I think Tandy told us earlier, any rate heterogeneity model is really a mixture model. If we think of the usual rate heterogeneity model with 4 discrete gamma categories, that really is just 4 GTR or whatever rate matrices multiplied by a scalar. So really all we have with a rate heterogeneity model is a mixture model where the mixtures take a very precise form of being scalars derived from this discrete gamma distribution. So just to show that we can get that back, we generated data on a random tree according to gamma rate heterogeneity, a continuous gamma with alpha 1. And then we analyzed the data using a homogeneous model, a gamma rate discrete gamma with 4 categories, and a pattern heterogeneity model with 4 rate matrices. And there's our input rate matrix, and there's the tree that we generated the 2,000 sites of. And here, if you fit the homogeneous model to the data, this is the time series plot of the apparently converged Markov chain. You never really know. It's part of the anxiety and fun of MCMCs. You never really know if you're converged. But here's the homogeneous model. And then here's the pattern heterogeneity model and the gamma rate model sitting right on top of each other. They get the same answer, as they should. And if you look at it a little bit more carefully, it looks as if the pattern heterogeneity model is doing a little bit better than the rate heterogeneity model, even though they're really the same models. That may be because this rate heterogeneity model is stuck with the 4 discrete gamma categories, and we generated these data from a continuous gamma, whereas this guy does a little bit better. He's not quite stuck on those 4 discrete categories. He can estimate the rate parameters any way he likes. Okay, let's have a look. So we can calculate from a discrete gamma what the input gamma transition rate parameters would be. That is, we take the rate matrix and we multiply it up or down according to the discrete gamma scalars. So you get a range of input transition rate parameters. And then here's the mean of the posterior distribution of the rate parameters we estimate from the mixture model. So you see that the mixture model can mimic very, very well gamma rate heterogeneity. Now that isn't surprising. It's just a way of showing that it can be very precise. It can find those signals in the data without you telling it it's there. And it can do that and get these things pretty well estimated. Now, because of that, and because we can deal with gamma rate heterogeneity, if indeed it is gamma with a single parameter, there's no point in fitting a pattern heterogeneity model and estimating 4 rate matrices when we can estimate a single parameter, that gamma scalar, that takes care of it. And so in all of the work I'll talk to you from now on, or describe from now on, any pattern heterogeneity that we find in the data is above and beyond rate heterogeneity. So we fit our models with gamma rate heterogeneity at the same time that we're trying to find these pattern heterogeneity. Okay. Now, one of the applications that we might think of for a pattern heterogeneity model is when we've got gene sequence data in which we expect different parts of the gene to evolve in qualitatively different ways. And secondary structure, ribosomal data, is often talked about as throwing up patterns of evolution that differ according to the secondary structure of the ribosomal sequence. So here is a data set of mitochondrial ribosomal 12S sequences in 57 mammals. And let's just have a look at the theory behind this a little bit. Remember that ribosomal sequences fold up into fairly well-known secondary structures. And there's some pretty good theory worked out about how the stems of these structures have canonical Watson-Crick base pairing. So G's always pair with C's and A's with T's and so on. And yet the loops of these secondary structures, there really is no theory about how they should evolve. And so what happens is that frequently with secondary structure data, people dream up models that are designed to deal with the stem evolution, these couplet models, I think they're called, because some very clear predictions are made about how stems should evolve, because they don't like to move out of this canonical Watson-Crick base pairing. And the conclusion that comes from thinking about stems and loops in secondary structure data is that you should see very high transition-transversion ratios in the stems, but no particular substitutional pattern is expected in the loops. Well, rather than partitioning up 12S data or ribosomal data of any sort into stems and loops, we thought let's just throw a pattern heterogeneity model at these data and see, first of all, how many patterns emerge from these data, how many qualitatively distinct patterns are in there, and do those qualitatively distinct patterns in any way correspond to stems and loops? Is our theory correct? So we did that. We fitted the typical gamma rate heterogeneity model to the data. with a GTR model with gamma rate heterogeneity. And then we fitted a pattern heterogeneity model with gamma rate variability in it, single parameter. And we discovered 4 rate matrices in the data and a substantial improvement in the likelihood. Now, when I say we discovered 4 rate matrices, I won't go into the technical details of that, except to say that in these cases, we fitted, a single GTR matrix, 2, 3, 4, 5, 6, testing at every step whether there was sufficient increase in the likelihood to justify adding an additional GTR matrix, and we tested those with Bayes factors. Okay, so we find 4 rate matrices, and the interesting question is, well, what do they correspond to? So here's our 12S data. Here's the 6 transitions in the GTR matrix. And remember that if we're talking about the usual sort of couplet model with stems and loops, we expect something with really high transition-transversion ratio, and then some stuff that we really don't know what to expect. And what we found was 4 qualitatively distinct rate matrices describe these data. This first one just looks a whole lot like a fast rate of evolution, sites that are just evolving rapidly, and there's a reasonable transition-transversion ratio. The second rate matrix is, or beg your pardon, this is the really fast one here, just things moving at a very, very high rate. The third rate matrix looks like our transition-transversion matrix. Here we have the C to T and the A to G going at a really, really high rate. So there's the expected transition-transversion ratio. Now remember, we didn't put these in. These emerge from the data. And then the fourth rate matrix looks like the very, very slowly evolving sites. And I should say here that frequently investigators will fit an invariant sites model to their data. We don't believe in doing that because we don't believe that sites are invariant. We believe that some sites don't show any variation within an alignment, but we don't believe they're invariant. We just believe they're going slowly, and we think that that matrix there is frequently the so-called invariant sites matrix. Okay, so we had these 4 distinct matrices, really led to a very large increase in the likelihood. But let's just have a look here now and ask, let's go back on a posterior basis and ask of every single site in the alignment, which rate matrix fitted you best? And in particular, let's ask for stems and loops, which rate matrix fits you best? So if we go to the stems, and we take our rate matrices here, we go to the loops, we take our rate matrices here, we can see that the first rate matrix looks like it sort of prefers loops. It's just kind of going along at a pretty good clip, and there's a lot of transitions, C to T. The second rate matrix really looks like the loop matrix. Loops evolve at a very high rate. They're just unconstrained in the secondary structure. The third rate matrix is meant to be our stem matrix, and it is a stem matrix in the sense that most of the stem sites are best characterized there. But look, 57 of these out of 180 or so of the loop sites are fitted best by that matrix. And finally, here's our sort of invariant sites matrix, if you will, fitting a vast proportion of them. What comes out of this is that any partitioning of these data into stems and loops is really way too simplistic, because some stem sites are far better fitted by a non-transition transversion matrix, these and these. 122 are best fitted by it, but the majority are not. And with the loops, it's just all over the place. And so the point here is that if you'd partitioned these data into stems and loops, thrown out the loops because you don't really know what to expect of them, fitted your couplet model to the stems data, using this transition-transversion matrix, you would have lost the fit for these guys here. And so we think that frequently the signatures and the patterns that are in real biological data are far more subtle and complex and interesting than the things we dream up a priori in our theories. Okay, well, that was an application to 12S data. I should tell you that when we do the same thing, frequently investigators divide up their data into codon positions, first, second, and third. We frequently find that such divisions, these partitionings, are much better served by a mixture model that doesn't divide them up, but simply fits the sites with the best matrix as it goes along. Okay, well, let's just talk about now a kind of larger scale application of the pattern heterogeneity model. We had 159 data sets sitting around in our laboratory just from various things we were doing, reasonable trees, typically quite small trees, but all pretty well sampled. And we dreamt up a reversible jump algorithm to kind of glue on to this pattern heterogeneity model. And a reversible jump Markov chain Monte Carlo is a wonderful technique dreamt up by Peter Green that allows you to run a Markov chain that can move to chains of higher or lower dimensionality. That is, you can actually change the number of dimensions in the chain as you're going along. And the reversible jump algorithm tells you how to do that. That is, you can run a single Markov chain that can move amongst various numbers of rate matrices, 1, 2, 3, 4, 5, 6. It just moves amongst those. And at convergence, it samples the posterior distribution of the number, in this case, of rate matrices that describe the data. And the jumps to higher or lower dimensionality are governed by the usual sort of acceptance term in MCMC, except here now, in addition to the likelihood ratio and the prior ratio, we have a proposal ratio that tells us how we're going to split rate matrices into maybe 2 matrices, or how we're going to merge 2 matrices back into 1. This is how we move into spaces of higher or lower dimensionality, and we need a proposal ratio term and a Jacobian, and this Jacobian is a term that sort of adjusts for the differing volumes of the dimensionality of these spaces. And there's some fun mathematics that you get to do to figure out these Jacobians. Okay, so we're going to be splitting or merging our rate matrices as we go along in this Markov chain, and then the Markov chain at convergence will sample the correct number of rate matrices to describe the data. So we want to run this algorithm on a whole lot of data sets and just ask how many rate matrices do you find, what do they look like, and so on. So the mean number of taxa in our data sets was about 40 with a big range. The mean alignment size was about 2,000 nucleotides, again with a big range. Here's just a kind of random selection of trees. Mean number of genes about 3 up to 37. We allow the RJ model or algorithm to run and just see what we get, really. Okay, well, the fun thing that comes out of this is here's the distribution of the number of rate matrices that this reversible jump algorithm finds in the data. You see that many of our data sets are described by a single rate matrix. A single homogeneous model would be an appropriate model for those data. These turn out to be the really small data sets in our sample. That'll be an important message to emerge. The modal number was about 2 rate matrices, and you can see that for some of our data sets it went up to 9 or 10 rate matrices were required to describe the data. Just very, very loosely, there's positive correlations with the number of taxa. The bigger the tree, typically the more rate matrices you find. So those signals are there. You just need lots of taxa to find them. It goes up with the number of sites in the alignment. It's positively correlated with the number of genes, all of the things you would expect. So great big data sets on very large trees are very likely to have lots and lots of pattern heterogeneity in them, and we probably want to be using something like a mixture model or a mixture model to characterize them. Okay, and then we have to estimate the weights of the various rate matrices, and so we can just ask what's the distribution of those weights, and you can see that the first rate matrix usually gets quite a high rate, and then it drops off, so 25, 30% or so, and keeps on dropping down. Okay, so we get a lot of diversity is really what we're saying here. In terms of these data sets, and bear in mind these are quite small data sets by and large, the mean improvement is about 265 log units, plus or minus 608, hugely skewed distribution. The mean improvement for data sets with greater than 1 rate matrix, though, because you can't improve on a homogeneous model if it is a homogeneous model. So our mean improvement when there was more than 1 rate matrix in the data was closer to 400 log units. And so there's substantial log likelihood to be picked up. And remember, this is all above and beyond gamma rate heterogeneity. And here's just a kind of plot of the improvement in the log likelihood with the number of rate matrices. And again, I don't want to make too much of this. It's rather obvious that the more rate matrices you have, the more log likelihood you're going to pick up. Seems to be increasing with the number of matrices. I don't know if that could be a confound with the size of the data set and so on. Okay, well, it's just some conclusions on this pattern heterogeneity model. It looks like we can detect it. It can lead, in general, to large improvements in the likelihood over a single Q model. It can improve the topology and reduce long branch attraction. I didn't show you any evidence for that, but in other work we've done, we've shown that we can get better topologies. And I think Hervé Philippe, who's got a mixture model for amino acid data, this is all for nucleotide data, has shown improvements or reduced long branch attraction. Mixture models can reduce the node density artifact. Those of you who are not familiar with this, the node density artifact refers to the tendency for us to underestimate the amount of change in long branches in trees. And mixture models, they just do a better job of characterizing gene sequence evolution, so they tend to reduce node density artifacts. Can be used to investigate gene evolution, such as secondary structure, or be applied to concatenated data sets. And I should also say that just in general, they do better in those 159 data sets that I showed you. The mixture model just blindly applied to the data improved upon the likelihood that the authors reported in their published work. Now, we weren't able to compare our work directly to theirs in every case because they didn't report exactly what they did, but when they reported a log likelihood, whether it was for a partitioned model or whatever, we could compare it to what we found with the mixture model. And I think in every case but two, the mixture model returned a better likelihood than these very carefully partitioned data sets that investigators had so painstakingly created. And I think it just shows you that we just don't know how to partition data very, very well yet. And we don't need to. Mixture models get around that. Okay, let's move on. That was the mixture model for heterotachy, for pattern heterogeneity. Let's move on now to mixture models for heterotachy. Heterotachy, remember, refers to different, or for a site evolving at a different rate in different parts of a tree. And this can be important because heterotachy has been shown to confound phylogenetic inference. We can get the wrong tree when sites evolve at different rates in different parts of the tree. And so it struck us that we might be able to develop a mixture model in branch length sets as a way of characterizing heterotachy. And we want to ask whether that model actually gives us anything worth kind of playing around with. Okay, so heterotachy might look like this. We have a single topology here. We think A is most closely related to B and C is the outgroup. But it might be the case that for some of the sites, they evolve rapidly on B, and other of the sites, they evolve rapidly for A. Here, those sites didn't evolve very much. Other of the sites might evolve a little bit in B, a little bit in C. So they're varying their rate of evolution depending upon where they're found in the tree. Okay, well, if you look at this, we have a single topology, 3 different sets of branch lengths. A homogeneous model is going to try to smear all these things together, but perhaps a mixture model that simply allowed, in this case, 3 different sets of branch lengths to characterize the data could pick up this heterotachy. And we want to ask whether that kind of model can work, whether it can improve phylogenetic inference, and whether we can fit the model to data and get reasonable results. So what does the model deliver? Well, we can get variation in rates across the tree, heterotachy. And we can ask here, maybe it's something like a non-parametric covarion model, because you'll all be aware that there is a model for variation in rates of evolution of sites across the tree, the covarion model, Tuffley and Steele. The covarion model is a very elegant model that prescribes a parametric switching rate in rates of evolution across the tree. All of the sites have to participate in that parametric distribution of switching rates. And what we're wondering here is maybe heterotachy manifests itself in a sort of non-parametric way. Okay. So the heterotachy model then, we find the probability of the data given some model of evolution now, Q. We'll just kind of hold that in the background here. But we're now going to sum the likelihood at each site over a set of branch lengths. So we have these discrete branch length sets, and these should be, are these in bold? They should be in bold. These are vectors of branch lengths, and we're going to sum the likelihood over those vectors of branch lengths, the topology being constant. Now, we don't put the topology in. We simultaneously search for the topology and estimate these branch length sets. Okay, does the branch length model work? Well, in 2004, when we first started working on this, there was a paper that caught a lot of people's attention. It was published in Nature by Kulikowski and Thornton, giving a case in which maximum likelihood was led astray by heterotachy. That parsimony outperformed maximum likelihood for these data. And what these two authors showed was that if you had some of your data corresponding to what they call this site pattern, that A and C, there'd been a whole lot of evolution, but B and D, not very much evolution. But other sites in your alignment, B and D had lots of evolution, but A and C didn't, so heterotachy, right? Different sites evolving at different rates in different branches. That in this circumstance, maximum likelihood would often return the wrong tree. That is, maximum likelihood would tend to put A and C together and B and D together, giving you the wrong tree. Okay, well, what interested us here was, well, could we solve this problem with a mixture model that estimated, in this case, two discrete branch length sets on the data? One branch length set would characterize that evolution, and the other branch length set would characterize that evolution. And would we get back the right tree? And would we, in this particular case, do better than parsimony, which was beating maximum likelihood? Okay, so we set up this 4-taxon tree simulation in which we simulate two sets of site patterns. Here, the one I showed you before, and then the one with B and D being long. 750 sites per pattern, so half of them simulated up that tree, half up that tree. We generate 5,000 random data sets from a pretty hard case where there's a lot of evolution along that branch, not very much at all along that one, or conversely, a lot along there, not very much along there. And then we vary the length of this intermediate branch, R, from about 0, that is a star phylogeny, to about 0.4. And then we analyze the data with maximum parsimony, Jukes-Cantor MCMC, because we just generated the data on a Jukes-Cantor model, the covarion model, the parametric covarion, and then what we're calling a branch length MCMC model, blind, in the sense that we're not telling it what the topology is or how many branch length sets are in the data. And the question is, do we get back the true tree, where maximum likelihood with these data tends to get back the wrong tree? It suffers from this long branch attraction. Okay, well now, with the reason the 4-taxon case is so hard is that there just isn't any information in the data. You can define very, very crudely something we can call the information ratio. There's 256 site patterns, right? 4 times 4 times 4 times 4 for A, B, C, and D. And the only ones that are informative as to the true tree give A and B the same nucleotide and C and D the same nucleotide, but differing from each other. So AACC, for example, is phylogenetically informative. And I think there's only 12 of the 256 possible site patterns that are phylogenetically informative for the 4-taxon case. So we can characterize each of our 5,000 data sets on what we might call the information ratio. How much information is there in the data as to the true tree? And we can see that when R, this internodal length here, is 0, there's almost no information about the true tree. But as R gets bigger and bigger, as we stretch R out, then you're more and more likely to get different nucleotides here from what you get here. And there's more and more information in the data. Okay, and so only when the information ratio comes up to about 1 here might we expect to get the true tree. That is, there's information actually about what the true tree is. That's why the 4-taxon case is so hard. There just isn't very much information in 4-taxon data. Okay, so there we have the information ratio creeping up to 1 or so, right around an R of 0.1. Now let's just have a look now at the performance of the various models in retrieving the true tree as a function of R. Okay, let's plot the R here. This is this internodal length here along the x-axis. And let's first have a look here at this curve here for the green and this blue. So here's the Jukes-Cantor MCMC with a single branch length set. This is just a standard Jukes-Cantor model fitted by MCMC, and the covarion. And you can see that we can calculate their P50 here, 50% probability of getting the true tree, and they arise to their P50 at an R of about 0.28. So when this thing's about 0.28, they will 50% of the time get the true tree. And if you go back here to 0.28, you can see the information ratio is about 1 there. Okay, so they're doing what they should do in the sense that when there's information in the data, they can get the true tree. Now remember, those are single branch length set models. Maximum parsimony does a little bit better. It gets the true tree with a slightly shorter r. So here indeed we're replicating this case that Kolakowski and Thornton published, that parsimony outperforms likelihood for this particular case. Okay, but look at the branch length sets model. Now here's where we fitted a mixture model on branch length sets, and we were able to retrieve 2 branch length sets in the data. And we were able to get a p50 for an r of about 0.039. And so the branch length sets mixture model is instantly finding that, yes, some of the data evolves according to this set of branch lengths, some of the data evolves according to this set of branch lengths. It can separate those two pretty quickly and get a true tree long before these other models can get it. And to give you an idea of how sensitive that can be for finding the true tree, if we go back here to the information ratio, and we plot the performance of the branch length sets model as a function of the information ratio, you see here's the probability correct p50 at right around 0.039. It's getting the true tree when there's a 50% probability that the data have got the right site pattern. Okay, so it looks as if mixtures on branch length sets can detect heterotachy in real data, and perhaps non-parametric heterotachy, and so improve upon the covarion model. And it looks as if heterotachy is a real problem in real data, and we shouldn't expect otherwise. I mean, we shouldn't expect all sites to evolve at the same rate, homogeneous throughout the entire tree. We should expect there to be a lot of heterotachy. Well, can we estimate sets of branch lengths? I mean, we did it for those simulated data. We were able to estimate 2 branch length sets. Here we simulated data up a tree of 60 tips. We simulated 2 branch length sets, and then we fitted 1, 2, 3, or 4 branch length sets to the data. And you see we get a big likelihood improvement over 1 branch length set when we've simulated 2. And if we add a 3rd and a 4th, there's not much improvement in the likelihood. Similarly, if we simulate 3 branch length sets up a tree, we get back 3 branch length sets, and we can estimate their posterior weights pretty well, about a 1/3 each, here about a 1/2 each. And again, there's no real improvement by adding a 4th. And similarly with 4, we get back the 4 branch length sets. And so it looks as if we can detect those bits of heterotachy in simulated data. But I think we want to ask ourselves here, this is a pretty crude and really sledgehammer to crack a nut technique, in the sense that here, if we're simulating data up a tree of 60 tips, to estimate an entirely additional branch length set, that's about 120 more parameters we've got to estimate on the data. And it may be the case that in real data, we don't need an entirely separate branch length set, but it may be that some of the branches of that tree are showing heterotachy and others aren't. And so wouldn't it be nice if we didn't have to put out a whole additional branch length set, but rather just estimate the branches that were showing heterotachy? And so once again, we go to reversible jump, Markov chain, Monte Carlo, and here we've dreamt up a reversible jump algorithm for heterotachy, in which we quite literally start with a tree and throw down 2 branch length sets that are identical. I'll say 2 here, but it could be 3 or 4 or 5 that are identical. And then we go to every branch in the identical branch length sets, and we ask, would you 2 like to be different in the 2 branch length sets? So we allow branches to split from 1 into 2, or merge from 2 back down into 1, or from 2 into 3, or from 3 back down into 2. So rather than estimating a whole additional branch length set, we're going to see if we can estimate just those branches that require us to add 2 or 3 or 4 different lengths to characterize the data. That is, how much heterotachy is there actually in the data. So we're going to allow each branch to choose 1 or more lengths. And now the topology is the same across all of these, but we're allowing the branch lengths to vary. So we need a mechanism for splitting and merging branches. Again, this is the job in Markov chain, Monte Carlo, or reversible jump of the proposal ratio. You need a mechanism for proposing changes, and you need to then adjust for the increase in the dimensionality of the space you're going to, and that's the job of this Jacobian. So you have to figure out all that stuff, which is really good fun. We've got that. and then we want to test this on some simulated data to see if it actually does anything for us. And so what we did was we threw down this random tree, and we generated 2 branch length sets on it, random branch lengths on this, so identical topologies, but random branch lengths. And so in theory, all of these branches could differ. When you plot the 2 against each other, they have a 0 correlation, just 2 sets of random numbers. But notice that, some of them, if you draw a 1-to-1 line up through here, some of them have got pretty similar branch lengths. And so maybe, under the reversible jump model, we don't need an extra parameter for things like that. 1 branch length will do for those because they're the same length. Okay, so we simulated data on those trees there. I think it was 70 tips on those trees. We simulated data to have 2 branch length sets, as I just showed you. And then here we're plotting the log likelihood returned by a 1 and 2 branch length set model. So here we fitted a single homogeneous model to the data, beg your pardon, with a single set of branch lengths. And then here the green is we fitted 2 branch length sets because we know there are 2 branch length sets in the data. We just threw the 2 branch length sets out there. And that's good fun. The green improves upon the red by, quite a large amount because we're characterizing the heterotachy. But the really fun thing is that with our reversible jump algorithm, we're getting nearly as much improvement. So the 2 branch length sets, which is the true model, improves by 925 log units over the homogeneous one, or the single branch length set model. But our reversible jump model gets 870 of those 925 log units with 73 branches rather than 138 branches. So roughly about half the number of parameters, we're getting something like 94% of the log likelihood. So that was encouraging to us. It looks as if we can run a reversible jump algorithm on this and maybe just be estimating heterotachy where there is heterotachy rather than forcing it all over the data. Okay, and here's a time series plot of the number of extra. This is just the posterior distribution of the number of extra branches, and it came in with a mean of about 73, and it just does the usual MC thing of wandering all over the place. And so here we're splitting and merging. We're moving up and down dimensionality in this space of the model. Okay, and then here's a eukaryote phylogeny in which we've applied, just because I said that we should just expect heterotachy everywhere. We just grabbed 48 eukaryotes off of GenBank that range from tiny little squidgy things like Trichomonas right up to very big important things like us. And we made this eukaryote phylogeny. I don't want to make any claims for this tree because, as you know, people spend their lifetime trying to make the eukaryote tree. But we just got 4 genes that are pretty well conserved off GenBank, drew this tree, and then we just asked if we fit our heterotachy model to it, do we see significant heterotachy in these data? So we have a 1 branch length set model. This is with gamma rate heterogeneity, of course. A 2 branch length set model here accounted for about 900 extra log units. That is, we just imposed 2 branch length sets on the data. And then we ran our RJ algorithm, which doesn't impose anything on it. And we picked up about 45 extra branches there. And we got about 96% of the log likelihood improvement back from 2 whole branch length sets. And so it looks like there's loads and loads of heterotachy in the data. And it looks as if we can pick it up with a sort of blind mixture model that doesn't know in advance where the heterotachy is or how much of it there is. Now I stress it looks as if we can because we haven't tested these things carefully with huge simulations. This is all we've done, but it looks as if it's working pretty well. Okay, so in summary, it looks like the branch length model works. One of the things that makes it very fun is that going back to an alignment, we can begin to say, well, in a gene, which sites are being speeded up, for example, which sites are slowing down. We can actually go back and say where in a tree and for what sites in the genes is heterotachy occurring. And then you can begin to link ecological conditions or physiological changes or morphological changes that are occurring along branches to changes in the selective pressures that are acting on genes and begin to take apart genes and say, oh, this part of the gene does that, this part of the gene does that, and so on. We can really, I think, pinpoint where in trees and which sites are responding in these ways. So I think this is a useful technique for kind of dissecting genes and the selective pressures acting on them. Okay, let's move on to the final topic, a mixture on topologies. All the same ideas now that we've been talking about for pattern heterogeneity or heterotachy, but now we're going to sum the likelihood over more than one topology at a given site. And the obvious idea here is that lots and lots of data have got conflicting phylogenetic signal. And I should tell you that the example I'm going to show you on a mixture of topologies is also one that Daniel Hewson and David Bryant have analyzed in a completely different way with splits tree, with splits tree. And I'll get onto that in a minute. But here the idea is we're going to sum the likelihood now of the probability of the data given some model of evolution over a number of independent topologies. We're going to allow the topologies to vary. And what are the reasons for this? Well, there's lots of reasons for conflicting phylogenetic signal, lateral gene transfer, gene duplication, where one gene then is lost, or goes extinct, can make taxa look similar that really weren't. So there's lots and lots of reasons to expect conflicting phylogenetic signal, homoplazy, convergent evolution, all sorts of things. So we should expect it in data. How does this model work? Well, here's the homogeneous model where we sum the likelihood over a single tree. The mixture model works by running a whole lot of trees in parallel, summing the likelihood over all of them, just like with the other mixture models. We're going to see that we can get then different topologies for different sites in the alignment. We'll do a simulation study in which we'll generate data up two different trees now. Notice that these two topologies differ. We're going to generate 20,000 random alignments. And we want to see how does the mixture model work when we allow the data to be characterized by more than one topology. So we've got these 20,000 random alignments of 1,000 sites each, where we vary the number of sites that correspond to that topology or that one. And you see if we fit a one topology model or a two topology model, we get an improvement. It looks like the two topology model is finding something. And in summary, we can sort of summarize the results by saying that when the proportion of sites in the data favor, 100% of them favor topology 1, the 1 and 2 topology models get back the same tree. When it's about a 50-50 split, some of the sites go for this topology, 50% go for that topology. The single topology model doesn't know what to do. It gets the star phylogeny, but the two topology model gets the two trees back. And when all of the sites favor the second topology, the two models agree. And so it looks as if it's not a bad thing to do. You always get the right answer. This is going to take a while to load. There's a lot of points here. If we just look here now at the proportion of sites favoring the first topology, and we follow the performance of the one topology model, it doesn't get posterior support for the first topology until it gets about 50% of the sites favoring that topology. And then as more and more of the sites favor it, it shoots up to having a very high support for that topology. If we fit a two topology model, though, it can start to detect that topology very much earlier because, and remember, it's fitting two topologies to the data. It's not forcing all of the data into a single topology. So the two topology model starts finding posterior support for the first topology far, far earlier than the one topology model. And up here, it doesn't seem to be doing as well, but remember, up here, some of the data are supporting a second topology. It's just the one topology model doesn't know that. This model does. And so it's never quite so confident as the one topology model. Okay, so it seems like it's a pretty sensitive thing. And let's just move on now and see whether we can use this topologies model to detect conflicting signal in real data. There's a long-standing problem in eukaryote evolution. It's called the ecdysozoa or coelomata hypothesis. Nobody really knows among arthropods, nematodes, and chordates, us, who goes with whom. And the ecdysozoa hypothesis suggests that arthropods and nematodes share a common ancestor. But the coelomata hypothesis suggests that Drosophila share a common ancestor with us. Now, this is important if you're a developmental biologist, because if you're studying Drosophila because you think they're a good model for studying human development, you better believe in coelomata. Okay, so we just collected a lot of data on these 14 genes, and we said, let's go and see if we can find conflicting phylogenetic signal in these data. Is that why this is so hard to nail down this tree, because there's just so much conflicting signal? And we fitted a one topology model to these 14 genes and whatever it was, about 14,000 base pairs. We fitted a one topology model to it, and we get strong posterior support for the Coelomata hypothesis, that is, putting arthropods, Drosophila, as a common ancestor with chordates, Homo sapiens. So we get strong posterior support from these 14,000 base pairs for the Coelomata. However, if we fit a two topology mixture model, now we don't tell it what the mixtures are, we just say, is there more than one tree here? We get quite a fun result. Here's our single topology model that I just showed you that gives this strong posterior support for Coelomata. But when we allow two topologies on the data, we get one topology with 100% posterior support for the Coelomata, and we get another topology with 100% posterior support for Ecdysozoa. And the posterior weights that we estimate for those two topologies are about 5743. That's why the total data set with a single topology returns Coelomata, but you can see those 14,000 base pairs hide two trees that are very, very divergent. That is, one strongly supports Coelomata, the other strongly supports Ecdysozoa. Okay, and we can just sort of wrap it up there. I think we're running out of time. So the summary on the topologies, again, it looks like the multiple topologies can work. It looks like we can estimate more than one topology and identify conflicting signal. Some questions are how to test such a thing, how many distinct trees are in real data, and then something I think Eric will talk about on Thursday is there seem to be some data sets that are compatible with more than one tree, and this is the identifiability problem. And this is a fascinating problem for mixtures on topologies. Okay, so just to wrap it all up, I mean, you've seen the kind of whirlwind tour of mixture models on pattern, heterotachy, and topologies. I just want to thank the people who pay for this work, the BBSRC and the NERC. Thank you.
So we have time for a couple of questions before the break. Yes.
What about the identifiability of your model?
Yeah, that's a very good question. The question is, what about the identifiability? We just haven't done that hard work yet. I don't know. I mean, the signs are, from the simulations, that it's pretty good. But that's why I mentioned the topologies issue at the very end. We know that one is not identifiable.
Could I ask, what about a label switching problem? You would have had to deal with that when you were saying, for example, whether a given